Before talking about my main subject, I would like to briefly review the EPR effect, which we proposed more than 25 years ago. This slide shows the normal tissue and tumor tissue, demonstrating the presence of a lymphatic duct in normal tissue and absence of any lymphatic duct in tumor tissues. Now, small molecules easily leak from the normal vessels, which gives us small molecules a short plasma half-life. On the other hand, the macromolecules have a long plasma half-life because they are too large to pass through the normal vessel walls, unless they are trapped by the reticuloendothelial system in various organs. In tumor tissue, it was found that solid tumor generally possess several pathophysiological characteristics, namely hypervasculature and secretion of vascular permeability factor, stimulating the extravasation of macromolecules within the tumor tissue, and absence of effective lymphatic drainage from tumors that impedes the efficient clearance of macromolecules accumulated in solid tumor tissues. These characteristics of solid tumors are basis of the enhanced permeability and retention effect, the EPR effect. The first subject is NC604, cisplatin incorporating micelle. NC604 consists of polyethylene glycol and the coordinate complex of polyglutamate and cisplatin. The particle size about 20 nanometer in diameter. This shows the nephrotoxicity of cisplatin and NC604 in rats. As you see in green column, 10 milligram per kilogram administration of cisplatin showed higher concentration of BUM and creatinine as compared to control and NC604. Then we conducted the phase 1 trial in United Kingdom, and we found that no renal toxicity occurs with minimum hydration, and GI toxicity was very weak. That is, almost all patients entered in this trial experienced grade 1 or weaker GI toxicity. However, we found that the high incidence of platinum-related allergy in this trial. At the moment, phase 1-2 study of gemcitabine plus NC604 is underway in patients with pancreatic cancer in Taiwan. Now, this is still ongoing. As you can see, as you can see, no renal toxicity occurred without hydration in this trial, and the GI toxicity was significantly reduced compared to cisplatin. Also, platinum allergy was controlled by intake of dexamethasone. From these results, we are considering that non-inferiority trial against cisplatin is feasible in various types of cancers. The next subject is NK115, paclitaxel incorporating micelle. The size of NK115 is about 85 nanometer in diameter, and clinical studies show that higher accumulation of NK115 in the tumor tissue and exerted potent anti-tumor activity. Then we conducted phase 1 trial, and then we found that the NK115 was well tolerated and the anti-tumor activity. As you see, significant reduction of liver metastasis was observed after the treatment in patients with pancreatic cancer. In this phase 1 trials, Cmax and AUC of NK15 is significantly higher than those of paclitaxel. The recommended phase 2 dose was determined to be 150 milligrams per square meter every 3 weeks. We then conducted a phase 2 study of NK15 in patients with previously treated advanced gastric cancer to see efficacy, safety, progression-free survival, overall survival, and others. As I said, NK15 was administered 150 milligrams per square meter every 3 weeks for 30 minutes infusion without any pre-medication, including steroid and antihistamine. Most common hematological toxicity was grade 4 neutropenia. Non-hematological toxicity was infrequent. Although NK15 was administered without any pre-medication, no patient experienced so-called acute infusion-related reaction. Also, it is noteworthy that during the whole period of this study, only one patient experienced grade 3 neuropathy, sensory neuropathy. It's 1.8% total. The incidence of grade 3 or 4 neuropathy of paclitaxel formulation, namely conventional paclitaxel, Zylotax, and Abraxane, were 10, 15, 11% respectively in each phase 2 setting. This anti-tumor activity, overall anti-tumor activity was 25% with 2 CRs and 12 PRs. Progression-free survival is 3 months. Overall survival is 14.4 months. The median overall survival in stomach cancer as a second-line therapy were reported to be 3 to 10 months. So this figure, 14.4 months, is remarkable. Now the conclusion, this first phase 2 study with NK15 at 150 milligram paclitaxel equivalent per square meter every 3 weeks provided positive proof of concept for high activity and tolerability of a new drug delivery systems formulation for paclitaxel. To clarify the survival benefit, a phase 3 will be evaluated. Now this table anti-cancer agent incorporated micelles under clinical evaluation. As I said, phase 3 study of NK15 will be started for breast cancer in 1 year. And SM38 incorporated micelle NK012 and cisplatin incorporated micelle NC604 is now underway. Also, we will start epilution incorporated micelle NC6 phase 1 study of NC6300. It's epilution incorporated micelle we started. That's all. Thank you for attention.